Welcome to lecture 13, the lucky 13. We're going to be talking about genetics today. And first of all, let's define what do we mean by genetics. What is the science of genetics? Well, genetics is the study of genes, how genes produce certain characteristics, physical and chemical, and how those characteristics are inherited, so how they are passed on. Okay, so just to understand what we're talking about, a gene, remember, is a portion of DNA that codes for a specific protein and therefore determines a specific characteristic or trait. Okay. Every gene has its own location in the DNA library, just like every book has its place in the library, every gene has its place in your genome. And every gene, as it has its specific assigned place, that place is called a locus. So locus, think of locus as location of a gene in the DNA library. Okay, so locus is a specific location of a gene on a specific chromosome. So last lecture, in lecture 12, you learned that we have 23 pairs of homologous chromosomes. Okay, so you have 2 of each kind of chromosomes, except for the sex chromosomes, which sometimes are homologous and sometimes they're not. So remember that you inherited one set of chromosomes from your mom and then the other set of chromosomes from your dad. So that means that if chromosome number 1, for example, contains a gene in a specific location coding for a specific trait, for example, a growth hormone, then you will find another version of that same gene on the mom's chromosome. Remember how I gave an example of the 2 recipe books on the same page, for example, on page number 157 in one recipe book, you will find a recipe for chocolate chip cookies. And the same page in the other recipe book will also be the recipe for chocolate chip cookies. So you're going to find the same gene in 2 different locations. Okay? And sometimes those would be the same or equivalent or equal versions of the same recipe, and sometimes they're 2 different versions. Okay, so you usually have 2 versions of every gene. One version came from mom, the other version came from dad. So the alternative versions of the same gene are called alleles. Alleles. So you have one allele, meaning a version of a gene, inherited from mom, and one allele, another version of the same gene, inherited from dad. Sometimes those alleles are the same or similar, and sometimes they're different. Okay, so sometimes they're the same, sometimes they're not. Okay, so it's just like 2 recipes. Those 2 recipes could be very similar or they could be very different. Okay, but you have 2 different versions or 2 different alleles coding for the same trait, coming from mom and from dad. Okay, all right. Now, let's, for example, take a look at, we're going to call it protein A, or protein X, whatever you want to call it. Let's say protein A is coded by gene A. Usually you name the protein and the gene with the same name. I'm going to call it A, letter A. So on mom's chromosome, we have one version of that gene. We call it allele A with the A capital. And the dad's chromosome, in the same location, the same locus, we have an alternative version of that gene. We're going to call it allele A lowercase. Okay? So if, for example, you have 2 different versions of this gene, an allele with the capital A from mom and an allele with the lowercase a from dad, then you have a combination, right? You have a version from mom, a version from dad. You have capital A from mom, you have lowercase a from dad. So if we were to state what alleles you have in every diploid cell of your body, we would have to name both mom's and dad's alleles that you inherited, right? So the list of the alleles you've got, the list of the alleles you've inherited, is called the genotype. Once again, the genotype is the list of all alleles present in an offspring. So whatever was inherited from both parents. Okay? When the gene versions or alleles get expressed, the result of that expression would be the phenotype. Okay, so genotype is what alleles you have. Phenotype is what would be the outcome of the alleles that you have. So the outcome of your genotype. So phenotype is the outcome of the genotype. Meaning genotype, it would be the cause, and phenotype would be the effect, the result. So the physical or chemical result of the expression of the present alleles. Okay, so the genotype is something that you cannot see. You can see phenotype. You can see what the person is expressing. You can measure it. You can observe it. But phenotype is a result of the gene expression. So that's something you can see. You cannot see what the genotype is because you can only guess what the genotype could be. But the phenotype being the result of the gene expression that you can see. Okay, so for example, your hair color or your eye color is your phenotype. It's a result of the expression of your genes. It's a result of your genotype. Okay, what alleles you inherited from your parents, your genotype, would dictate what you can observe, your phenotype. Right? If your genotype is different from your sister's or brother's genotypes, then you will probably have different phenotypes. Okay? So this way you and your siblings may have different physical features. You may have different height, different body type, different hair or skin color, different shape of nails, different eye color. So different genotypes means different phenotypes. Okay. So what we're going to talk about next is what determines the specific phenotypes. Okay. So it's not always very simple. It's not always straightforward. Okay. It's not just about different versions of the same gene that you've inherited. It's a little bit more complex than that. Because phenotype actually depends on two things. It depends on the genotype, which means the list of the alleles that you got present in every cell of your body. And the second is how those alleles interact with each other. Okay? So what alleles you've got and how they interact with each other. Okay? So let's use one trait so that we can discuss the genotypes and phenotypes. It's just easier if we do it with an example. So we're going to use the eye color as our example. Okay, so there are two major versions of the gene coding for the pigment molecules found in our eye's irises. Okay, your eye's iris is the colored part of the eye, and you have blue and brown pigment molecules that you could express. You could express blue or you could express brown. Okay, so let's say we're gonna use the small b for blue and the big B for brown. Okay, we're gonna use a a lowercase letter b for blue color or blue allele, and the uppercase b for brown color allele. Okay. So when we record the genotypes, if you have a brown eye color, then most likely for brown eye color, an allele with a capital B would be the one coding for the brown. So capital B codes for brown. The lowercase b codes for blue. Okay. So when you can record the possible combinations of what you could inherit and what would be the outcome. So possible combinations of mom's and dad's alleles and the genotype in the offspring. So if you inherited a little b from mom and a little b from dad, then your combination would be two little b's, right? That's your genotype. Your genotype would be little b, little b. If you inherited a big B from mom, a big B from dad, then your genotype would be big B, big B. If you inherited one big B from mom and little b from dad, then your genotype is big B, little b. And then if you inherited little b from mom, a big B from dad, then your genotype is again big B, little b. It doesn't matter which one you put down first. What matters is the list, what's present. Okay? So now let's take a look at all these genotypes and figure out when they get expressed, these alleles get expressed, what would be the phenotype? Okay? So you probably noticed that in some cases, like the first two cases, when your genotype is little b or big B, big B, okay, the two alleles you got are the same. In that case, that type of a genotype, we call it homozygous. Homo for the same, zygous for a zygote. So think of a zygote as the first diploid cell that you started from. So homozygous genotype is when you have two alleles that are the same. When you have two alleles that are different from each other, for example, big B, little b, then we call that genotype heterozygous. Hetero means different, zygous as in zygote, and so that means that you have two alleles that are different from each other. Okay, so if we go and determine phenotypes based on that, well, it's pretty simple and it's pretty straightforward. If you have two little b's and each little b codes for blue, you're obviously going to express blue color of your eyes. If you have two big B's, you're obviously going to express brown color of the eyes. However, when the situation is heterozygous, when you have two different alleles that you've inherited, then you have to look at how these alleles interact with each other. Because in a homozygous genotype, the phenotype is pretty straightforward. In a heterozygous genotype, you have to look at how the alleles interact with each other. Okay, so for example, if you have big B, little b, the brown versus blue, well, blue is much lighter color than the brown. So brown seems to be louder than the blue. So the brown kind of dominates over the blue. So that brings us to understanding the next point, is that some alleles will act as dominant alleles. They will be louder and brighter, and they will be expressed very clearly. The recessive alleles would be the opposite of the dominant. The recessive alleles are much softer and quiet. They kind of stay quiet. If the dominant allele is present at the same time as a recessive, you will see the dominant being expressed, you will not see the recessive being expressed necessarily, because dominant is way louder than the recessive. Okay, so I'm giving an example here that if you have two people in the room and both are whispering, you'll probably hear them both. If you have two people in the room who both are speaking loudly or yelling, then you will hear them both. But if you have two people in the room and one is whispering and the other one is yelling, you're going to hear the one who is yelling, right? So even though they're both there, you are only noticing the one that's loud. Okay, so both alleles, dominant and recessive, are expressed when present. The dominant allele masks the effects of the recessive one. The recessive allele is overshadowed by the dominant one. Okay, now understand very importantly that recessiveness of an allele has nothing to do with the quality or the value of the gene or of the allele. It gets expressed, it's just not noticeable. Okay, when the dominant one is being so loud, you just don't notice the recessive one. It's there, you just don't see it. Okay, all right. So what we need to understand is this: when we have homozygous genotype with both alleles being dominant, we call that genotype homozygous dominant. Both alleles are the same, they're both dominant. In case of the eye color, homozygous dominant genotype would result in brown eyes for the phenotype. In case of a homozygous recessive genotype, so homozygous recessive means homozygous means two alleles are the same. Recessive means they are both recessive, and you would see blue eyes for the phenotype. Now, when you have a genotype where you have two different kinds of alleles, one dominant and one recessive, then you just call it heterozygous genotype. You don't say heterozygous dominant or heterozygous recessive, you just say heterozygous. So when it pertains to regular autosomal genes, and it has nothing to do with codominant genes, which we're going to talk about later, when you have heterozygous genotype, then you are going to see the expression of the dominant allele. So you're going to see brown eyes in the case of eye color. Okay, so please note that the recessive alleles are always expressed when present, but can result in a recessive phenotype only if you have homozygous recessive situation. So homozygous recessive. So the only time you're going to see blue eyes is if you have homozygous recessive genotype, meaning both alleles code for blue eyes. Okay. Also note that we can always predict a phenotype from a genotype, but we can tell the genotype from a phenotype only when you have homozygous recessive situation. Okay, so when you look at someone and see blue eyes, you know for sure that they have little b for their genotype. Okay, so next, you're going to practice solving some genetic problems. Let's go over some basic genetic problems. When we're solving genetic problems, there are, I mentioned here 2 steps, but I'm actually going to say 3 steps. There are 3 steps. First step when you're figuring out genetic problems is usually in a genetic problem, you are given a situation and you're given the parents' either phenotype or genotype. So you need to figure out what are the genotypes of the parents. And you want to write that down. The next step is you're going to draw the Punnett square. The Punnett square is this box where you're going to put the alleles, you're going to separate the alleles of one parent horizontally and the alleles of the other parent vertically. And then you're going to combine them inside the Punnett square to look at all the possible combinations of the parent's alleles. So you're going to be basically copying from the top, from the side, from the top, from the side, from the top, from the side, and from the top, from the side to look at all the possible combinations of these alleles. And when you have all the possible combinations of the alleles, then you can go on to step 3, which is not mentioned here in the lecture. Step 3 would be to interpret the results, meaning translate the genotypes that you're going to see in the Punnett square. These are the genotypes of the babies, of the offspring. So translate the genotypes of the offspring into the phenotypes. So figure out what would be the phenotypes based on these genotypes. Okay, so we are going to try to do it with the eye colors. Okay. Over here you have some examples with alleles little a, big A. But let's just go ahead and do that with eye color. Okay. So for example, here. So you have a mom. Here, let's take a look at this problem over here. Problem 1. Mom is heterozygous. Dad has blue eyes. The question is, what is % chance of them having a blue-eyed baby? Okay. Now, it doesn't matter which parent you're going to put horizontally, which parent you're going to put vertically, but it's very important that we understand the steps. Okay, I'm going to start using my pen here. So mom is heterozygous. So hetero means different. So that means she has 2 alleles and they're different from each other. So that means that it's going to be like this. Big B, little b. There. Dad has blue eyes. The only way to have blue eyes is to have 2 recessive alleles like that. Right? So that's step 1. Step 1 is you establish the genotypes of the parents. And always write it down. Have a piece of paper in front of you. Don't let your head hold the information. Let the paper hold the information for you. It's so much easier. When it's in front of you, you can see it very clearly. So when you have established that, you're going to put, for mom, we're going to put mom at the top. We're going to put one of her alleles over one column and the other one of her alleles over the other column. And we're going to do the same for dad. We're going to put one of his alleles for one row and the other one of his alleles for the other row, like that. So here's mom's big B, little b, and dad's little b, little b. Okay. Now, once we have done that, from here it's pretty straightforward. We just copy from the top, from the side, from the top, from the side, copy from the top, copy from the side over here, copy from the top, from the side. So now we have established the possible genotypes of their babies. These are their babies, okay? And now that we have all their genotypes, we can figure out their phenotypes based on the genotypes. So let's take a look at their phenotypes. So this one, this baby, is heterozygous, like mom, right? And because the brown is dominant over blue, then this baby is going to have brown eyes. This baby, same thing, okay? So this baby is brown-eyed. This baby, however, has 2 little b's, so that means this baby is going to have blue eyes. And this baby is going to have blue eyes. So 2 out of 4 possible babies would have blue eyes. Okay, 2 out of 4 will have blue eyes. And it's asking us, what is percent chance of them having a blue-eyed baby? Well, 2 out of 4, that's 50%, right? Half, 50% chance of having a blue-eyed baby. And that's your answer. And that's basically how you solve all the genetic problems. You figure out the genotypes of the parents, you draw the Punnett square, figure out the genotypes of the babies, and then finally you interpret them. Okay, let's move on to the next part. Okay, here you see the results of it. And then you get to practice doing these with the practice questions. So make sure to get plenty of practice with these. And then you're going to have some extra credit assignment available doing some genetic problems for extra credit. Okay. In the meantime, okay, after you have done your extra credit or you've done your practice, we're going to go on to the X-linked genetic problems. All right. So last lecture, we talked about the X-linked genes. Remember, the chromosomes numbers 1 through 22 are called autosomes. The chromosome number 23, the chromosomal pair number 23, are called the sex chromosomes. Okay? In some people have 2 X's for their pair number 23. Some people have an X and half of an X, or a Y, as we call it, for their 23rd pair. And if you have 2 X's, that means you're a female. And females, because they have 2 X's, they have two alleles for X number 23. Males have only 1 allele because they only have 1 X number 23. The other chromosome codes exclusively for maleness, so it's not going to help us too much. Now, please note the genes that are found on the X number 23 have very little to do with the female sex, actually. The femaleness, the code for femaleness is actually spread throughout the genome. And if you remember, the embryo would develop as a female from the get-go up until 10 weeks of gestation, when if there is a Y chromosome present, it would kick in and the baby would detour its development from female to male only in the presence of the Y chromosome. Okay. So the Y chromosome codes exclusively for maleness. The X chromosome does not code for femaleness. Okay? The femaleness is coded throughout the genome. Okay? So a female, because a female has 2 X chromosomes, will always have 2 versions of every gene found on those X chromosomes. So even a female who carries an abnormal allele on one of her X chromosomes will usually have a backup copy on the other X chromosome, on the second X chromosome. So females usually have a backup plan, okay, kind of like plan B. If one X chromosome lets you down, well, then you have the other X chromosome. For males, that is not the case because males don't have a backup plan. They don't have a backup copy of that X chromosome. They only have one, and the only one. Okay? So when we are talking about X-linked genetic disorders and X-linked genes, when we do the X-linked problems for these genetic disorders, we use the X's. Okay, we put an X with an N for normal, which would designate a normal gene found on the X chromosome. And then alternatively, we would put an X with a different letter like C for color blindness, or H for hemophilia, or D for dystrophy. next to that X to signify an abnormal X chromosome. Okay, one of those X chromosomes gets passed on, and then you would need to look at what could be the outcomes for the offspring. Okay, so if you have, for example, a female who has a normal X and a normal X, you're going to have a healthy female. If you have a female who has one X normal and the other X that is affected with the mutant gene, then this female would be only a carrier of the abnormal allele, but because she has a second X chromosome that's normal, she has a backup copy, then she's not going to express the disease. She's going to be healthy. So both XN, XN, meaning X normal, and X normal and X affected, in both cases, the female would be healthy. It's just if she has one of her X's being mutant, then she is a carrier of a mutant gene, but she's not sick, she's not affected. If she has two X's that are affected, well, that's just bad luck. It's extremely rare. I actually haven't seen it, but I guess it does happen. A healthy male would be the one with an X normal and a Y. Remember, Y cannot be normal or abnormal. Y only codes for maleness, so you're not going to put any letter next to it. For the X that is affected, that would be the only X that the male would have. So X affected or X mutant with the Y chromosome would be the genotype of a male who's affected by the disease. Okay, so you need to understand how genotypes of the X-linked genes translate into phenotypes for these males and females. Okay. All right. So again, when we're solving X-linked genetic problems, same steps. Step number 1, we have to figure out the genotypes of the parents. Step number 2, we're going to do the Punnett square. And step number 3, we have to interpret the results. Okay, so let's take a look at one of these problems and practice solving them. Okay, so let's take a look at this one. We are looking at the X-linked genetic disorder called hemophilia. Okay, hemophilia is a genetic disorder when the X chromosome, the chromosome number 23, that contains a gene coding for clotting factor number 10, has that gene mutated. And because that gene is mutant, when that gene gets expressed, you're not producing a good working clotting factor number 10, and as a result, you cannot have your blood clot properly. So that means when you bump into things and you bruise yourself, or you get a paper cut, your blood does not stop. It keeps going and going, and it keeps bleeding, and you could actually bleed out very quickly. Okay, so it's a very serious problem because a person can bleed out very quickly and very easily from like the smallest wound. All right, because the blood does not clot. It doesn't stop the bleeding. It doesn't stop the losing of the blood. So we're going to look at the problem when mom is a carrier and dad is healthy for this X-linked genetic disorder. Let me get to my pen again. Okay. And if mom is a carrier, now first of all, mom is a female, so that means automatically 2 X's. Okay. Now she's a carrier, which tells us that she is healthy, but one of her X's is affected. So that means that one of her X's is normal, and the other X is hemophilic. Okay. Now dad is healthy. Now dad is a guy, male. So it's an X and a Y. Okay. Now only the X can be normal or hemophilic. And since he's healthy, his X is normal because he only has one X. He cannot have two kinds. Okay, so now that you have established the genotypes of the parents, you can draw the Punnett square. Okay. Now, same thing, you're going to separate mom's and dad's alleles. So mom, we're going to put one of her alleles over one column and the other one of her alleles over the other column. So we're going to put XN over this column and XH over this column. And then for dad, same thing. We're going to put one of his alleles here. Okay. And the other one of his alleles over here. Okay. And we do the same thing. We copy from the top, from the side, from the top, from the side. So if we go and copy from the top, from the side, from the top, we have XH at the top, so we copy that. And then from the side, we have XN. And then same thing here, XN from the top. We copy Y from the side. And XH from the top. And the Y from the side. Okay, so let's take a look at the outcomes here. Okay. Now, if we take a look at what we have here and interpret that, let's take a look at what we have here. We have an XN. First of all, it's a girl because she has two X's and both her X's are okay. Now we have XNY. That's a boy and he's also healthy. Okay. Now, this one is interesting case because it has XH and XN. So one of the X's is affected, but the other one is not. So we have a girl who is a carrier of a bad gene, but she's still healthy. So this girl would be healthy. Okay. And if we look at the last scenario, we have an XH and a Y. So that would be a boy with a hemophilia. So that one would be sick. Okay. So that would be the last step, interpretation of the result. So 3 out of 4 would be healthy, and 1 out of 4, which is 25% chance, 1 out of 4 would have hemophilia. Now, if I asked you a question differently, if I said that this man and this woman, they are having a baby, they're pregnant 20 weeks, they're going to their ultrasound and they want to find out the sex of the baby, and they find out that they're having a boy. Do they need to worry? If they find out that they're having a girl, do they need to worry? Okay. If they know that they're having a boy, then they know that they could have either this boy or this boy. 50% chance that the boy could be sick. If they're having a girl, they don't need to worry because they know no matter what kind of girl they're going to get, she's going to be fine. Even if she's a carrier, she's going to be healthy. Make sense? Okay, so you're going to practice solving genetic problems like this. So try to do these practice problems, and then you also have some extra credit problems here to solve. And in the meantime, we're going to move on to codominance. Okay, so what is codominance? Let's talk about that. So, I mentioned that sometimes you have two different alleles and two alternative versions of a gene, and sometimes one is more dominant and the other one is recessive, right? So sometimes you have two alleles where one is really loud and the other one is very quiet, right? So in some cases, you can have two alleles that are equally loud. Okay, so if you imagine two people singing in the room, but they sing in different voices, you would hear a duet. Okay, so instead of one whispering and the other one is speaking loudly or singing loudly, you would have two who are singing loudly, and yet they have two different voices. So you would hear both of them and you would hear a duet, you would hear a combination. So for example, when you have, let's say, I am giving you an example of a flower, right, that is expressing some red color and expressing some white color. Okay, so if you imagine, once again, if you imagine two people, okay, when we're talking about dominant versus recessive, one is singing loudly and the other one is very, very quiet, you're going to hear the one who is singing loudly. And if you have a different situation where you have two people who are singing loudly, but they're singing different voices, you're going to hear a duet. It's going to be a combination of the two expressions. And here I'm giving you an example of a flower that is expressing two different alleles. One allele is coding for a red color pigment, and the other one is coding for a white color pigment. So if one plant inherits two R's for red, the flowers will be red. The other one inherits two W's for white, the color would be white. But if a plant inherits both R and W, both red and white, then it's going to express both red and white, and the result would be a combination of the two, and you're going to see pink flowers. Okay? So we're looking at combinations of equally dominant alleles being expressed. So when you have alleles who are equally dominant, both dominant, and when they're present, they're both expressed to the same extent, we call that codominance. So co means together, dominant. They're dominant together. Okay. Now in humans, we can see codominance in blood types. Okay. So what are we talking about here? Let's take a look. So everybody has red blood cells, right? Everybody has red blood cells in our blood. And those red blood cells are produced in our bone marrow from stem cells. And so that means that they all have the same DNA, and they should all be like identical twins of each other. They should all be the same. All our red blood cells should be the same. So every cell in your body has these little ID proteins, and red blood cells also have ID proteins. And we call those, we have two different versions of ID proteins. We have version A and we have version B, as we call them. So a version A of an ID protein, if you have an allele, meaning a gene, in your genome coding for protein A being expressed on the surface of your red blood cells, then you will express protein A on the surface of your red blood cells. And we'll say that you have blood type A because you have A protein on the surface of your red blood cells. if you have a different version of that gene, a different version of that allele, alternative version, version B, in your genome, then you will express that protein B, and you will have blood type B, because you have B protein on the surface of your red blood cells. Now, some people inherit both allele A and allele B. Now, because A and B are both dominant, they will both get expressed. And as a result, you will express both A's and B's on the surface of your red blood cells. And we say that you have blood type AB. People who express neither A nor B, so they don't have allele A and they don't have allele B, so they don't express neither A nor B. They have red blood cells with neither A nor B. They have 0, or we call it blood type O. And here O stands for 0, nothing. No A's, no B's. Okay? So we need to clearly understand what genotypes could code for these phenotypes. So when you have blood type O, it means that you have genotype O and O. So that means you have inherited nothing from mom and nothing from dad. If your blood type is AB, it means that your genotype is AB. You have heterozygous codominant genotype. Now, because nothing is recessive to something, then when you are expressing A's or B's, they will always be dominant over nothing, over 0. So that means that if you have inherited A from one parent and nothing from another parent, you will express the A. If you've inherited B from one parent and nothing from another parent, you will express B. All right. So let's take a look further here. So blood type A. Okay, blood type. If you have blood type A, remember blood type is a phenotype of which is a result of a genotype. So to have blood type A, you probably either have inherited A from mom and A from dad, from both parents, right? Or you could inherit A from one parent and nothing from another parent, because presence of the A protein dominates over the absence of anything, or a protein. The presence is dominant over the absence. Because you have an A, you're going to express an A, all right? So you basically have, if you have an AA genotype, we say you would have a homozygous genotype with blood type A. If you have AO genotype, then you have so if you have a heterozygous genotype AO, it means you inherited A from one parent and nothing from the other parent. You still have blood type A. So you have heterozygous genotype with blood type A. You could have homozygous genotype with blood type A, or you could have heterozygous genotype with blood type A. So when you are doing a genetic problem on codominance, you have to carefully read what kind of situation you have. Okay? The same thing with blood type B. If you have a homozygous dominant B type, then you have BB. If you have heterozygous genotype with a blood type B, then you have a BO genotype. Okay? So the same steps apply in codominance problems. You still determine the parents' genotypes, you do the Punnett square, and then you interpret the results. So if we do this, so mom has blood type O, dad has blood type AB. Okay, so what are their genotype? Well, for mom to have blood type O, the only way to have blood type O is to have a genotype of O and O. O. Blood type AB is possible only if you have genotype AB. That's it. Okay. What is percent chance of having a baby with blood type A? So pretty straightforward. We do the Punnett square. Now that we have parents' genotypes, mom, dad, okay, and all we do is just copy from the side, from the top, from the side, from the top, from the side, oops, that's O, from the side, from the top, from the side, from the top. Okay, so let's take a look at the phenotypes we got here. So this baby has genotype A, O, so it will have phenotype blood type A, because remember O is recessive next to an A. Same thing here, blood type A. Over here we have blood type B and blood type B. So we have two babies of blood type A and we have two babies with blood type B. Okay. Pretty straightforward, right? So it's asking what's percent chance of blood type A baby? That would be 2 out of 4. That would be 50%. All right. So once again, it's very important that you read the problem and see what it says. Okay. Let's take a look at another one. For example, if we say that someone is heterozygous codominant, what would it be? The heterozygous codominant. So that means heterozygous means two alleles different from each other, and codominant, together dominant. So that would be A and B. That's the genotype of a heterozygous codominant person. If we say someone has heterozygous with blood type B, okay, so to have blood type B, you have to have at least one allele saying B, and the other one has to be either a B or a nothing. Now, if it was a B, then it would be homozygous, but the problem says if they're heterozygous, so that has to be an O. Okay, does that make sense? Or if we say that someone is homozygous with blood type A. Homozygous with blood type A, that means that they have one A and the other allele has to be also A because they are homozygous. So it has to be like this. That's their genotype. Make sense? Okay, I'm going to erase this. Okay, and I'm going to let you practice answering these questions and practice doing more problems like that. And when you get a chance, please take a look at this video on heredity. And when you're done with that, we can go on and talk about eye and vision. Now, since today we talked about eye color, it kind of makes sense that we mention the anatomy of the eye. Okay? And the basic things, you just need to know the basic anatomy of the eye. And I suggest that you just take this picture and just redraw it a couple of times and label it. Once you redraw and label everything, you will memorize it for a long time. Okay, so if we look at the eye, it has the eye has the structure of a ball, and the vitreous body is the inner space of the eyeball, which is filled with fluid. The innermost layer of the eyeball is called the retina. It's black. And the retina is the part of the eye that contains the photoreceptors. Photoreceptors, meaning detect light. Okay? And when they detect light, the signals get sent to the optic nerve to the brain to interpret those signals. Okay, then the next layer around, right behind the retina is called choroid. Choroid is this red colored layer. It's right behind the it's right behind the black retina. If you notice somebody, if the light hits their pupils just so, their pupils will appear, instead of black, they will appear red. Also, you would see that on pictures sometimes when you get a red eye effect on a picture. What happens is if the light hits the retina so strongly that it actually goes behind the retina and it hits the choroid layer. Choroid layer is red because it's very vascularized. Then the retina, which is kind of see-through, even though it's black, it's kind of see-through. So through the black, you see the red choroid behind the retina, and it's red. Okay, and the outermost layer of the eyeball is called the sclera. Okay, the sclera, and the front of the eyeball is called the cornea. Very thick layer that's is supposed to protect your eye. Okay, the sclera and cornea is very strong, thick layers. Very, it's cartilaginous, so it's white in color, and that gives the eyeball its white color. Okay. Now, let's take a look at the front of the eye. So this structure over here, kind of like in front of the iris, this is called the aqueous humor. Aqueous humor. It keeps the lens, which is this structure over here. This is the crystalline lens. The crystalline lens allows you to see objects at a different distance depending on how stretched that lens gets, and it gets stretched by little muscles over here. These little muscles are supposed to control the thickness of the lens, and these are called ciliary muscles. You can see where it says right here, where the yes, it does. So the ciliary muscles, they regulate the thickness of the lens and how round the lens is. So the lens could be changed between more round or more flat, and that would allow you to focus on objects farther or closer from the eyeball. Okay. Over here you have a window into the retina. That window is a pupil, and the window is regulated, the size of the window is regulated by the iris. The iris is this colored muscular portion that basically makes the window bigger when your pupils dilate or smaller when your pupils constrict. Okay? So iris controls the amount of light that would enter through the lens and hit the retina. Okay. All right. In the very front of the eye, there is a very thin film-like membrane covering the very front of the eye. Okay, it's not the same as cornea. Cornea is this thick layer, but very thin membrane is called conjunctiva. Okay, the conjunctiva contains the blood vessels that you can actually see when you look at a person's eye and you see the little tiny little blood vessels. Those are in the conjunctiva. They're not in the cornea. They're in the conjunctiva, in that very thin mucousy film. Okay. So let's talk a little bit about the eye retina. So we mentioned the retina, which is this black innermost layer of the eyeball, contains the photoreceptors. The photoreceptors detect light, and they allow you to see images when your brain interprets those signals. And these photoreceptors come in two types, the rods and the cones. They are named after their shape. So the rods, they look like this. The rods are used in dim light, so at night when there's not a lot of light, when it's like dark in the room, that's when you use your rods. Rods use vitamin A or retinol to get activated in the presence of a dim light. You have many more rods than you have cones. So rods are more numerous than cones. They don't allow you to detect color, but they allow you to see in dim light. And there's only one kind of rods that you have. And they produce kind of low resolution blurred images at night. So if you, next time you get up at night to get a glass of water or go to the bathroom, just pay attention how everything looks around at night, how your room looks around. Can you detect colors? Can you see colors, or do you remember the colors? Can you really see the colors? Probably not. It's really hard to tell the color of something in the dark. You can see the shapes of things. You can see the shadows and the shapes of things, but all the images are a little bit blurry. They're not very sharp, and all the images are of the same color, kind of like the shades, different shades of gray. Okay. Now let's go back to the cones. The cones, again, are named after their shape, and you don't have as many cones as you have rods because they don't need as much stimulation. See, with the rods, you use rods in the dark, and you need to use a lot of rods to produce images in the dark because there's not a lot of light. And you also need to use retinol, vitamin A, for your rods to get activated, because in the dark, those rods get, in the rods, retinol, which is vitamin A, gets converted into rhodopsin, and the rhodopsin stimulates the nerve endings in that optic nerve. Now, the cones work differently, and you don't need as many cones as you need rods, and you have 3 different kinds of cones, and thanks to that you can detect different colors. You can see all the colors of the rainbow because of those cones. They are less numerous, they detect colors, and you have 3 kinds. You have red cones, blue cones, and green cones, and you produce high resolution images when using those cones. And of course they are used in bright light, so when the sun comes out or when you turn on the light, you can see all the colors. If you can see all the colors, that means you're using your cones. If you don't see colors and you only see the shades, you're using your rods. Okay. Now, next part I want to talk about is what is macula and what is fovea. The macula is a special place in the retina where you have a very high concentration of photoreceptors, high concentration. And you use macula to see images and be able to focus on images. Fovea is this indentation in the macula where you have a very high concentration of the photoreceptors, and you use that spot for reading, watching TV, looking at things and noticing details in the images that you see. Like, for example, right now you're either watching this video, and you can either read or at least see the images here. So you're using the fovea in your eye retina to see all these images. Okay. Last but not least, extrinsic and intrinsic eye muscles. The extrinsic eye muscles would be the muscles you would find on the outside of the eyeball. That's why they're called extrinsic. They're outside. So these muscles are used to simply rotate or move the eyeball so that you can look in different directions. The intrinsic eye muscles would be inside the eyeball. So these would be the ciliary muscles that control the lens, the thickness of the lens, and the iris muscles, which would be over here on this picture. The iris muscles, they control the dilation and the constriction of your pupils. So you need to know all these terms that are highlighted in red font and all the terms that I have mentioned here. And make sure to watch this quick video. It is pretty quick and short. And there's another video on the physiology of the eye and how it works. And have fun with it. Make sure to do all the practice questions before you take the quiz today. All right. Have a great day. See you next time.